Hello students, in this video I will let you know about enzyme. It is a topic of chapter biomolecule. Basically when you are going to know about biomolecules, what do you mean by biomolecules? Basically biomolecules are all the molecules which are present in the living system, in the living cell. Basically all the carbon containing compounds that is present inside a living system, that is basically and involved in the metabolic reactions, either it may be catabolism or anabolism, that will be called as biomolecules. It is further categorized under two categories on the basis of their molecular weight, that is micromolecule, and the second one is the macromolecule. In macromolecule there is macromolecule, that is a protein. Basically proteins are made of amino acids which are linked to each other with a peptide bond, and there is formation of peptide chain and forms protein. As we know, proteins are of different structures. It may be a linear chain, it may be a helical chain, it may be a helical structure, it may be a tertiary structure, it may be a quaternary structure. So when we need to know about the enzymes are something that enhance the rate of reaction by their catalyzing property. And what is that? Whenever a enzyme helps the substrate, helps the reaction to increase the rate of reaction, they decrease the activation energy. And activation energy is something that is a barrier for a reaction. Whenever it needs to cross, the enzymes are required, and the reaction occurs. So basically in this I will let you know, for example, this is the enzyme, this is the enzyme, and most of the enzymes are of tertiary structure, 3 degree structure and helical structure. Not helical, they are the tertiary structure. Just like as we know, the primary structure is like this one, where each amino acids are linked with each other. Similarly, like this one, this is the linear chain, the primary structure. In the secondary structure, it is the helical structure, just like a rotated ladder. In the second case, the tertiary structure, there are many loops. During the formation of many loops, there are certain services or the pockets, and these pockets are this one. Basically, this is called active site, where the substrate binds, okay. And the second and the fourth category is the quaternary structure. When we will discuss the protein, I will let you know. So basically this is the substrate. For every biochemical reaction, there is a requirement of enzyme. Without enzyme, rate of reaction, in some case the reaction will not start, and in many cases, in almost cases, the rate of reaction is very slow. Just like I will let you know example of carbonic anhydrase. Basically carbonic anhydrase, it is very much required in the combination of carbon dioxide with water during its transportation in the form of carbonic acid. Whenever the CO2 combines with water, it forms carbonic acid. If this reaction occurs without the enzyme, the rate of reaction is very slow. By listening, you will be amazed, you will be surprised that 200 molecule per hour. This is the rate without enzyme. If we place the enzyme, if we provide the enzyme, the rate of reaction increase many folds, 1 times, 2 times, and 3 times, not 100 times, it is 10 million times. Means when we provide the enzyme carbonic anhydrase for the formation of carbonic acid, it will increase the rate of reaction by many folds, that is 10 million times. And how much? basically the rate of reaction becomes 6 million molecule per second. So you can measure the difference. It will be approx, yeah, more than 10 million times. So the substrate fits over the enzyme to catalyze the reaction to increase the rate of reaction by decreasing the activation energy. I will let you know, but for this moment, for this instant, I will let you know that uh, enzyme is something which decrease activation energy and increase the rate of reaction. And activation energy, the minimum external energy which must be provided to start the reaction, to initiate the reaction, to overcome the reaction. That is the it is a barrier which must be broken down, which must be crossed, so that the reaction can occur. Okay, so basically enzymes are the organic biocatalyst. As we have heard about catalysts are the thing, or the compounds, or the ions, or the substance which increase the rate of reaction in non-living system. So there are two types. The first one is the bioorganic catalyst, organic biocatalyst we can say, and second one is inorganic biocatalyst. Inorganic catalyst. Inorganic catalysts are required in the non-living reaction, and in the non-living reaction. In case of living system, living reaction, there is a requirement of organic biocatalyst. So basically enzymes are the organic biocatalyst which increase the rate of reaction by decreasing the activation energy. So let's know about these things in detail. So the definition of enzyme is the organic biocatalyst which increase the rate of reaction by lowering the activation energy. This we called as enzyme. Okay, if there is a question that who coined the term enzyme, so basically we have a scientist whose name is Kuhne. Kuhne is a scientist who coined the term enzyme. Basically the word enzyme, there is two words. In means in and zyme means yeast. Means the isolation of this substance, this colloidal substance. Basically enzymes are the colloidal substance which are made up of macromolecules, basically protein, and proteins are further made up of amino acid, which are the micromolecule. Okay, so Buchner is a scientist, Buchner, Buchner, who discovered and isolated the enzyme from yeast cell. That enzyme, the name of that enzyme is the zymase from the yeast cell. In next we have a scientist Sumner, who in next purified and crystallized, means in the form of solid, the enzyme urease from a plant, that is the jack bean, and this plant is also called as Canavalia. Basically here we have zymase enzyme isolated and discovered from the yeast cell. The term enzyme was coined by Kuhne, and the enzyme in first of all purified and crystallized by Sumner from jack bean, which name is the jack bean, and the scientist name is Sumner, and the enzyme is the urease. Okay. So one more thing I will tell you that the on the basis of their catalytic power, the fastest enzyme that we must know is the carbonic anhydrase, whose rate of reaction is very very fast. Means the rate of formation of molecule is 6 lakhs molecule per second. In next, the slowest enzyme is the lysozyme, whose number is very very slow as compared to the carbonic anhydrase. Okay, it may be, it is the rate that basically 30 molecule per minute. Okay, so the fastest enzyme is basically, and the slowest enzyme. The fastest enzyme is carbonic anhydrase. Carbonic anhydrase. And the slowest is the lysozyme. If we compare, you must know lysosome, lysozyme, and their rate for formation of molecule is 6 lakh molecule per second, and in case of lysozyme it is 30 molecule per minute, means half molecule per second. This is the rate. It's very half molecule per second. How much we can say? How much is the difference between the such kind of enzyme? 30 molecule per second. Okay. So this is all about their comparison in the fastest as well as the slowest enzyme. But what is the criteria to whom we will call that it is the enzyme or it is not the enzyme? So there are certain criteria which the enzyme must fulfill. Like we can say all enzymes are protein. All enzymes are protein, as I earlier told you that enzymes are made up of mostly tertiary protein. So we can say all enzymes are protein except ribozyme. It is not lysozyme. It is the ribosome. It is basically ribosome, the protein factory, which synthesize the enzyme ribozyme. Clear? Ribozyme is the enzyme which is formed from the nucleic acid. Means we can say which nucleic acid, RNA or DNA? It is the RNA having catalytic power and catalyze the reaction. That's why this is the non-proteinaceous enzyme made up of nucleic acid, that is ribozyme. And these enzymes are basically synthesized, basically these are a protein. So protein factory is the ribosome. Under genetic control they synthesize protein. Okay. So let's know about the characteristic of enzyme.